Dear listeners, welcome to the first episode of the Science Podcast about enzymes and molecules. I am Roberta Foresti, Professor of Biochemistry at the University of Paris-Créteil and working as a scientist at the Mondor Institute of Biomedical Research in France. I have been working for most of my scientific career on a specific enzyme called Heme Oxygenase, investigating the action of this protein in health and disease. So it seems natural that the first episode of this podcast will be dedicated to this subject. Today I'm excited to welcome our first guest, Dr. Leo Otterbein, a longtime collaborator and friend. Leo Otterbein, PhD, is Professor of Surgery and Principal Investigator of a lab at Harvard Medical School in Boston, USA. Dr. Otterbein has published fundamental findings on heme oxygenase and its product, carbon monoxide, establishing the crucial role of this system in inflammation. One of the primary goals of the Otterbein Lab is to study the benefit of carbon monoxide in low doses to help against organ injury, for example in the heart, kidney, or lungs, and trying to understand the mechanisms that drive this protection. In this respect, Dr. Otterbein has led a major effort in developing approaches to deliver carbon monoxide to organisms for therapeutic application, including a recent gas-entrapping material that will be discussed during our conversation. Today, Dr. Otterbein and I will talk about some aspects of heme oxygenase and carbon monoxide biology, including their main function, the toxicity versus the beneficial activities of carbon monoxide, as well as its impact on inflammation. We hope you'll enjoy our deep dive into this metabolic pathway and that you will learn more about this research field. So hello, Leo. Thank you so much for being with us today.
No, it was a pleasure, and thank you for the wonderful and very kind introduction. I'm glad to be here, and I'm eager to discuss the science that you and I both have interest in.
Fantastic. Thank you. So, Leo, to set the context for our listeners, let me start by asking you, what is heme oxygenase and what does it do?
That's a very good question, and I think most textbooks will tell you that the heme oxygenase system is designed to catabolize or metabolize heme, and of course heme is present in many forms in the body, and intracellular, as well as it floats around in the bloodstream as well. And the enzyme is designed to catabolize the heme into three products. And most of the time, those products are considered as just that, products. Carbon monoxide being one, iron is another molecule, an atom that comes out. And then, of course, there's the biopigments that more people are familiar with, such as bilirubin, which is one of the most potent antioxidants in the bloodstream. So many labs were settled on identified when the enzyme was identified to say this is the limits of its purpose, just to catabolize heme. But in the early 90s, the enzyme sort of regained some of its a lot of interest because it was shown to be very protective when it was increased. And some seminal papers came out that showed that if you can increase the enzyme's expression and therein the activity, that you could get very beneficial effects in multiple models of disease. So it became of very big interest through the 90s and into the 2000s, where those like myself have more raised the questions of why does this enzyme serve its purpose, such as protection, more than just heme catalysis? And as you alluded to, how are the products involved in some of the mechanisms that people observed with the enzyme itself? So it's had an interesting history. It's gone from a textbook metabolic enzyme to one that is coined a heat shock protein, and eventually to a protective gene. And now many labs are still trying to figure out exactly what it does and how it works.
Well, especially because we know that we have a constitutive heme oxygenase and an inducible one, which is, overexpressed during stress situations. So there is obviously a meaning in that. Why don't we just use the constitutive one, which is there all the time, and why do we need to upregulate another enzyme which basically does the same catabolic activity? But
yes, that is a fundamental, excellent question, and I think one that I don't have the answer to as to why evolution designed two enzymes. Presumably, there's different purposes for each. As you mentioned, one is constitutive. It's been told and been published that the HO2, if I speak to that one first, is primarily localized to the brain and the testes, but I think that more recent literature would suggest it exists elsewhere. The HO1 is as you mentioned, also is an inducible form, so it's not present basally in most cases, although there are systems, for instance, in the spleen where HO1 is constantly expressed. So that's the place where red cells go to die and the heme is metabolized continuously. I think the evidence that really opened the field was when they created the knockout mice. So when you create these genetically engineered mice that don't have the HO1 and also those that don't have the HO2 enzyme, then you begin to see what the purpose and the relative role is. So the HO1 deficient animal, and there is an HO1 deficient human correlate, both agree with one another, the mouse and the human, that if you don't have this enzyme, you are at a very large disadvantage in terms of dealing with stress. There's also an underlying phenotype that these animals have and the human have that I could talk about more, but I think in contrast to that, the HO2 deficient animal is otherwise normal phenotypically, and there are incidences where that animal can be shown to have some effects, particularly if you challenge it in the brain. So why there's two, I think that's a great question. I don't have the easy answer for it, but one might argue that there's a backup system in case one does fail.
Yes, yes. So you were saying that one of the products of heme oxygenase and heme degradation is carbon monoxide. So we are saying that in our body, we have an enzyme that produces carbon monoxide, but we know that carbon monoxide is a silent killer. It's a poisonous gas. So how can we explain this paradox?
This is another excellent question, and it's one that I actually start out when I give lectures to students or to anyone. I raised this question, and it really comes of interest when you talk to those who still believe, and rightly so, that carbon monoxide is a poisonous gas. And the question or the statement that I always raise is, if this enzyme generates carbon monoxide and it generates carbon monoxide during stress, and these are stresses that might be otherwise lethal. There are instances where heme oxygenase is induced with kidney or liver injury, diabetes, neurological disorders, where you produce more of this gas. And the question that I raise, and no one seems to have the answer yet, is why would the body create a gas that is dangerous, one, and two, why would it generate a gas in more concentrations and higher levels when otherwise the organism is at risk? So it doesn't make any intuitive sense. Now, that being said, there is a level, as with every substance, oxygen and water being included, where that substance becomes toxic. And so there is a limit on, just like watering oxygen, carbon monoxide will achieve lethal levels. So I'm one of many, including yourselves, that pioneered the idea that low levels of carbon monoxide, maybe not so much equivalent to what's generated by the enzyme, would have a biological effect. And that's what we've spent the better part of 25 years trying to understand, is why this gas has essentially two different faces. But everything has two different faces. There's a toxic level, and then either a physiologic level, or in this case, maybe even a beneficial purpose, evidenced by the animals that don't have HO1, that they can't generate carbon monoxide. And there you see very detrimental pathophysiology.
Yes. So it looks like the body can produce carbon monoxide, at the same time can contain the ranges of carbon monoxide in order for them not to be toxic. So this is also very...
It's the only gas, I'll just add one thing, it's the only gas, the only substance, if you will, that is not metabolized as it's generated. So it doesn't participate in any other reactions. It's produced by the enzyme and is eliminated as the same gas from the lungs. So it's an interesting molecule in and of itself, unlike some other products that get further metabolized or altered and handled differently by different organs. It's an interesting gas in and of itself in that regard. So it's generated a lot of interest, at least for our research.
Yes. So staying on carbon monoxide, you have your research has mostly focused on carbon monoxide. And in one of your original findings, you discovered that exposure of animals to low concentrations of CO protected the lungs against hyperoxic injury, that is, injury caused by very high oxygen levels. can you tell us why you thought of exploring CO among the different products of heme oxygenase, and why and what stimulated you to look at hyperoxia in the lung?
So the reason why I had spent a number of years in a research lab as a technician, I hadn't decided to go into professional school yet, studying heme oxygenase 1 in collaboration with a couple different investigators who were interested in the enzyme. And the question at that point was, we knew that we could induce the enzyme and it was very beneficial. It begged the question at that time, why was the enzyme, or how was the enzyme protective, and whether it was mediated by one of its products. And since then, the enzyme has had a lot of different paths that it's taken, and things like the enzyme can regulate gene transcription, and it has other effects in the body and the nucleus and other cytoplasmic locations. But we asked the question at the time, whether one or more of the products would have a biological effect. And iron was one that we ruled out early because it was very well described that the iron was chelated by the iron storage proteins ferritin. We spent some time studying the biopigments, and they were just technically hard to work with. So I happened to be in a research lab just by chance that was interested in gases. And we were primarily focused on oxygen toxicity and ozone toxicity, which were, in a public health scenario, are gases that, not so much oxygen, but certainly ozone, are certainly at the forefront of lung injury. And we knew that lots of oxygen, which is known clinically, is dangerous to the lung and the rest of the body. And of course, ozone, most are familiar with, is something you try to avoid because it's very reactive and cause tissue damage, cellular damage. So I was very comfortable with working with gases. So I approached one of the engineers at Johns Hopkins where I was working, and said, I want to create an environment of carbon monoxide, and here's how I'd like to do it. And fortunately, I found the right person at the right time, and we created the exposure apparatus that was necessary, which was very similar, if not identical, to what you would use to expose animals to oxygen or ozone or any gas. We were doing other gases, sulfur dioxide, for instance. And so we tested it. I went to the literature, as everyone does at the time. I was beginning my PhD, and found that there was levels that were tolerated, and made the assumption that we could get low enough that might equate what's produced by HO1. And simply because the oxygen was a familiar area for me at the time, and I was working with it, I knew that it caused lung injury, and there were very good measurements of injury that I could adapt that to the carbon monoxide study. It was probably to your audience, probably the worst possible thing I could have chosen to study is oxygen, because as everyone knows probably, that oxygen is the treatment for carbon monoxide poisoning that they use in the hospitals. We can talk about that, of course, and what that means. But essentially, I was giving the antidote to what I was trying to study, which was very very much challenged by most who asked me why I chose oxygen. So your question brings back fond memories of why I chose that as a first go. It turned out that it was helpful because the carbon monoxide was protective in the lung. And then I quickly expanded that to other non-oxygen, if you will, non-antidote giving. It'd be like studying a bacteria and giving antibiotics. It just wasn't logical at the time, but I was young and foolish, and maybe I chose incorrectly, but it did work out, fortunately. And so that's the story of how carbon monoxide and oxygen came to be the first study, and how we initiated those kinds of experiments.
Yes, very interesting. I think, although I don't think you pursued that, and not many people have pursued that, the fact that probably carbon monoxide and oxygen have similar bindings, have similar binding proteins, may come into the story, although we really didn't study that very much, but it's probably very important in trying to understand how the carbon monoxide is working in the presence of oxygen, because obviously there will always be a competition between the two gases for oxygen.
That's exactly right. And I think in hindsight, you could say it let us understand where CO might be binding. And we've since concluded, and I think your work would argue that too, is that the places where oxygen goes, carbon monoxide can go as well. So there's a very delicate balance between these gases that I think focuses us on where it might be acting and how.
Yes. So if we continue a little bit with your story, which is the story of heme oxygenase, subsequently you made another fundamental discovery showing that CO exerted anti-inflammatory effects in macrophages. And in this work, you clearly demonstrated how CO could inhibit the production of pro-inflammatory molecules stimulated by bacterial particles, bacterial lipopolysaccharide, such as TNF-alpha and IL-1 beta, and inducing at the same time the expression of anti-inflammatory molecules such as interleukin-10, for example. I think this work really set the basis of our understanding of HO1 as a multifunctional enzyme, an enzyme that is capable of both fighting oxidative stress and inflammation. Do you think that these two roles are connected?
I think that what we've learned, initially you described what we were observing in the animal. So I'm a physiologist by training, so I always take my experiments right to the whole organism, in this case mice or rats or pigs. But I think we learned a lot from the oxygen experiments. We quickly moved those, as you mentioned, into endotoxin experiments, where we looked at the effects of CO on inflammation. And for many years, we dissected that to say, how was it working as an anti-inflammatory molecule? And I think it's important to note that what we eventually came to the conclusion was that carbon monoxide kind of fits the need, or HO1, if you will, if you can equate those. And I don't want to equate those too much, right? We tend to equate HO1 endogenous CO production with what we give exogenously. And to your listeners, I want to make it clear that we don't really have evidence that actually happens. We see a lot of similarities, and they may in fact be true, but what we see with HO1 may be very different mechanistically from what we see by giving exogenous CO. And one example of that, or another example of that, would be where it regulates inflammation. I don't talk about it much more as an anti-inflammatory. It does have those properties, but if you were to put, in this case, a live organism in there, like a live bacteria, you begin to see a different phenomenon happening. So one would argue that if CO is anti-inflammatory, that in an active infection, for instance, it would be a very deleterious event, right? Because now you're disabling the immune response to deal with the bacteria. So in fact, if you give carbon monoxide before a bacteria infection, you can actually do some harm, because you've conditioned the animal, in this case, to have an anti-inflammatory phenotype. And now it doesn't have the apparatus, if you will, to help fight against the bacteria. If you then shift that experiment and give the carbon monoxide during the active infection, and it's important because you would never give something before an infection, because no one knows when they have an infection or not until that happens. So giving it before it was irrelevant. But if you were to find somebody that was sick with a bacterial infection, it would be after the fact. And if you give carbon monoxide then, you actually amplify the machinery in the immune cell, in this case, the cells, the macrophages and the cells that fight the infection. You amplify that. And we think that that's a different mechanism of action, because what happens is in our studies and in our conclusions is that as the cell changes its phenotype due to activation states, such as when you give bacteria, different proteins show up. Different enzymes, for instance, that were not there before, now become very nice targets for carbon monoxide, and oxygen for that matter. And the example I always use, which is one we've studied forever, is nitric oxide synthase, which is an enzyme that has a heme, which is a target for CO. So to come back to your question, it's an anti-inflammatory molecule for sure. It isn't always an anti-inflammatory molecule, because in the case of bacterial infections, it's actually pro-inflammatory by helping kill the bacteria. And it may just be that it has different targets in that setting. One question that's always come up, I want to get back to more questions you have, is if the body can increase HO1 and the cell can increase HO1, why do you need more? And that's a question that everyone asks. And if you have a very astute student, they should ask that question right away. They should say, Dr. Foresti or Dr. Morlini or Dr. Otterbein, HO1 is induced by these stresses. Why do you need to give more? And the answer that, of course, comes up is it's just not enough, that you've overwhelmed the system such that now the pathology takes over and you can no longer handle the infection, or in this case, and you would argue that the evidence lies back in that knockout animal, where those animals can't manifest any response to the infection. And so they actually will die at a much, much weaker dose of bacteria than a normal mouse will. So it becomes a threshold, if you will, that where the natural system, the endogenous system, just can't compete and can't maintain the or correct the problem. And now you have to give extra. And that's where our studies and your studies and others have come to say giving more at that point is beneficial.
Yes. Also because in this respect, we have to think about also the substrate. We never knew exactly where the substrate was going to come from, whether there was enough substrate for the conversion into carbon monoxide. And this is why maybe sometimes in particular situations, you do need the extra addition of CO coming exogenously. Yes, but
Excellent point. And that's very true. So we're assuming a lot with this enzyme that it has what it needs to do the job it's designed to do.
yes, because if I think about situations like diseases like sickle cell disease, where we have an increase in endogenous heme levels because of continuous hemolysis, and we know that there are, for example, macrophages that keep patrolling the blood vessels, and they sense where there is the hemolysis occurring, and they take up the heme and they metabolize and they sense and they help clear up the vaso-occlusions, for example. So, maybe there are also localized situations where we may have a lot of CO, but in a certain, scenario, in a certain localized, organ or part of the organ and so on.
Yep, exactly.
In your efforts to understand the mechanisms that underlie the protective activities of CO, you identified MAP kinases and JNK phosphorylation as being modulated by CO, suggesting that the signaling activities of this gas interfere with the classical signal transduction cascades of the cell. You also show that mitochondria and HIF1 are targeted by CO. So, we still have a doubt about what is really the target or the targets of CO, because we know that this gas, as we alluded to before, has a high affinity for metals. And we also know that these particular proteins, such as the MAP kinases of HIF1, they don't normally contain metals. So these findings, in my opinion, emphasize how, in reality, we know still very little about the mechanisms that the cell employs in order to sense CO. So what do you think is the preferential target of CO? And if you had to put a kind of, sequence of events, once the cell is exposed to CO, what would be the sequence? Where is CO binding? What happens next? And so on.
Another great question. And I think there we have to turn to what we know about the potential targets. And there's some interesting literature, more reviews of late, looking at if you were to rank order, to use your system or your question, to rank order what might CO bind to preferentially. And it's interesting because the classic targets become that are more, quote, popular, if you will, like the mitochondria. Guanylate cyclase is another one that is very popular in terms of literature that speaks to them as the targets for CO. There are proteins that no one really explores yet that have actually even a higher likelihood of being bound by CO. The number one that I've seen and been told is something called neuroglobin, which exists in the brain, and no one studies this protein. So, or doesn't study it in the context very much so yet with CO, to my knowledge. And so I don't know whether we really understand the relative affinities and whether biochemistry will tell us, and the physical chemistry, if you will, of whether that is logical to say that CO will bind to any metal or any heme, if you will, in this case, to use that as an example. With the MAP kinases, of course, they don't contain any target for CO. I'm told by physical chemists, though, that anything can bind to anything. That's a very general statement, and that CO could bind very weakly, albeit, to any protein, if the right conformation and the right environment were present. The mitochondria became the obvious target because that's what literature and dogma tells us, is that that's where it poisons the cell. We know from some other studies and others as well that the mitochondria begin to generate reactive oxygen species in response to CO, and that in of itself I don't like the term oxidative stress and reactive oxygen species because I don't think we really understand them. I think we just use them as in many cases, as a catch-all, if you will, to say this is how it works. It's because it's oxidative stress and reactive oxygen species do this and that. I don't mean to downplay those who study reactive oxygen species. Very important. I don't know if they exist long enough in a cell to really have the effects that we see versus other potential explanations. But modulation of things like kinases, while not direct targets for CO, become indirect targets for CO through things like, one could argue, reactive oxygen species generation, changes in the oxygen tension, which would argue why HIF might be changed, that the oxygen sensors in the cell become disabled or are confused, if you will, by the fact that CO is now bound and there's not ability to have oxygen present, because typically carbon monoxide will bind stronger, more affinity for those targets versus oxygen. But again, I don't know if we know enough about the relative molecular affinities of these proteins and these enzymes, if you will, for CO versus oxygen, and whether they even are legitimate targets at all. One paper that came out said the guanylate cyclase is the weakest, has the weakest affinity for carbon monoxide. Yet there's studies that suggest that it's important in vasomotor tone. So I think there's still a lot to learn. I think that no matter what you do to a cell, what you measure in a cell, this is what I tell some of the students I work with, is you'll find something that changes and you can chase it and make a story out of it. Does it give us any more insight into the exact target? Maybe, maybe not, but chances are good if you want to see some change, you'll see it, and CO will probably change it one way or the other, but it may not be the exact target that you're looking for. So still a lot of work to do.
Yes. And maybe, as you were saying before, it may depend also on the circumstances of the cells, because we know that HIF1, for example, is activated in low oxygen levels. So, as you were saying, in this particular situation, you may have the induction of genes and proteins, expression of proteins that will become sensitive to CO, and whereas normally you wouldn't have them. So really, I think it's very important to define very well the situation, the scenario, your experimental conditions, and probably we would need the help of pure biochemists.
100%.
Yes.
I know two in Paris that are very excellent biochemists. But you're right, and I think that's there's a lot still to learn about this system and what the targets are and how the cell changes in response to CO. Is just still much to learn.
Yes. So you've used CO gas exposure. We talked a little bit about this, which was also tested in clinical trials. You have used CO releasing molecules in their various forms and demonstrated their utility in preclinical models of disease. A very recent addition to this work is your recent publication on gas-entrapping materials that can be foams, hydrogels, or of solid composition, where CO is encapsulated. Can you describe what these substances are and what they do, and what do you see as an advantage in the use of these molecules, these new substances, compared to the previous CO delivery systems that you used in the past?
So the story with the gas-entrapping materials, which are termed now GEM, are one that was pioneered out of a series of bioengineers at Massachusetts Institute of Technology, MIT. And I was fortunate to be introduced to someone over there who was a materials type of scientist, the first author and the senior author, who are remarkable drug development and formulations engineers, if you will. They think about these things all the time. And in a collaborative conversation, we discussed the methods of CO delivery. And to their credit, they said, why don't we create a foam of some nature that had yet to be defined and use that as a delivery system, because you can create a scenario where you can pack gas molecules in a very high concentrations into a material that will then allow it to be released as you will. And these are concepts that are above me in terms of how they make these formulations, if you will, so they can make the components such that they release carbon monoxide very quickly. They can make them so they release carbon monoxide very slowly. the other aspects that are innovative potentially is that you can then give a trigger, so you can add something like EDTA that will force the release of, so you can time when you want to release them. So these guys created this and then had no way to understand how to test it in animals and preclinical models, hence my role. They said, we think we've created this, but we don't have a way to test it. Can we come to your lab and evaluate it in your models? And that's really where the collaboration was cemented. And the idea was that you could deliver these. They're created, as is sort of alluded to in the paper, with essentially the term we use is gastronomy. These are agents that they use in things like making cappuccinos, or they use them in making fancy desserts at restaurants. And these are no more than that. These are gastronomical ingredients that you can essentially assemble in a way that you can infuse them with any gas. And in restaurants, they use nitrogen or CO2 or something else, or oxygen maybe, or maybe just air, room air. And they said, this is simple. Let's just make a version or versions where we can just impregnate or incorporate the gas, the CO. And so they created it, we validated it. And the uniqueness of them is that they can be changed very simply by changing the materials that they're contained in. So the solid form is actually a candy that doesn't get a lot of press in the manuscript, but essentially they show the solid version of that. And that is sugar. That's just gas entrapped in sugar. All it is. And if you put that into your mouth, or you put that we put it into the cheek of a mouse, it'll dissolve just like a piece of candy. And as it does so, the carbon monoxide is released. So it was a very unique way to deliver and administer it, and it overcomes some of the challenges that were being faced with inhaled, with some of the small molecules, even with some of the liquids that are still being investigated, because it allowed you to tune the amount of CO rapidly with what they call generally regarded as safe ingredients, GRAS reagents, which are a term that the FDA, Food and Drug Administration, use to define agents that they are already accepted as safe, and you can use them for anything you want in terms of human administration. So we applied these in models in the paper, in models of intestinal inflammation, inflammatory bowel disease, colitis, if you will. And then we also wanted to know if there was a reason why it would have effects given in this case, as a intrarectal delivery, whether you could target organs that were beyond the bowel. And that was why we did the liver studies. What their utility is still to be defined. There's limitations, of course, on how much you have to eat in order, if you were going to take it orally, how much of this you would have to eat, unless you like to drink a lot of cappuccino. You know that foam is not always a lot in terms of how much you actually take in. So the utility might be something as far as topical delivery, perhaps. And that's been looked at. There's some ongoing work looking at how well it heals wounds and things like that. So you could apply it as a part of a bandage after a surgical procedure or an injury in a car accident, or there's any kind of open wounds, maybe you could apply it that way. So still a lot to be learned with that and where it might be clinically useful, but that was the premise of the origins of GEM.
Yes. So the solid form, where you say that it was basically like candy, did it have enough CO to be measured when you give it to the animals, for example, or I mean, this is a consequence of what you said. You don't know if people would be willing or capable of really eating or whatever these big amounts of these solid forms, for example. Do you think this is one of the limitations of the?
I think the challenge is going to be, how much CO do you need? How much is going to be enough? And that's been a question that's plagued the field forever, is how much do you need to have to get a biological effect? And I think there's differences there between what you use as your preclinical evidence. Some of these models that exist, and there's many, of course, you may only need a small amount of carbon monoxide daily to get an effect, whereas in some more severe situations, such as ischemia reperfusion injury of an organ, you might need to reach high levels very quickly. And so there's a lot of work being done to say, what is the real therapeutic index, the level that you need in any given scenario. And I think it's going to differ depending on the disease indication you're choosing. As far as the solid, it would sort of speak to that. If you were going to use it to treat something that required a low amount of CO, then it might be very amenable to a dosing regimen, or you could put that piece of candy in, I'll use that term loosely. I don't know what it tastes like, but I think you can make it sweet, that you could get your dose just by letting it sit in your mouth. There's evidence that you can do sublingual delivery of nitric oxide, which they use for people with unstable angina of the heart, and it goes under the tongue, and it's a drop of liquid that contains enough agent to create dilation of the vessels. So I think the jury is still out, if that's the best way to term it, to say that there's utility there, at what dose and how to deliver it. So I think there'll be different uses for different levels of administration. So certainly the small molecules are going to have their advantages. The inhaled gas, I think, has seen its day, if I might say that. I think there's that's a whole nother podcast we could have about the history of that and how that went. But I still think there's a lot of room, and I think there's still hopefully some group will evaluate this efficiently and effectively in a clinical trial that will demonstrate whether it's truly beneficial or not, like we've seen in so many different animal models.
Yes, the solid form, starts to really remind us of a medicine, of a drug,
right,
that could be taken. Just one last question before we finish. Do you think we can use these molecules in a targeted way? Maybe it's easier to do it with these molecules compared to some other forms of CO. Certainly we cannot do it with a CO, a gas exposure. But maybe with these molecules, we can advance also in that direction.
Yes, I think some of the work certainly that your labs have done speaks to that possibility, and whether you can target, I think, is a very important avenue of research. I think if you can get the CO, the one common thread here, of course, is that anyone you speak to, as you well know, hears the word carbon monoxide, they want to avoid that. And the clinicians, I can tell you that their biggest concern is what is the amount of CO in the blood. So they measure that as carboxyhemoglobin. And if there's a way to eliminate that component altogether, so you don't even see a level of carbon monoxide appear in the blood as carboxyhemoglobin, and yet you get a therapeutic benefit through targeting, that's the game changer. That's the winner to me. If you can find the right molecule or the right combination of molecules that will allow you to target it. There's been talk of making a folate receptor targeted molecule where you can say, I can deliver specifically, for instance, the kidney. There's another one, I can't think of it, that would specifically target the heart. So if you could deliver your payload of carbon monoxide to that cell in that organ and concentrate it, much like we started this out by saying maybe HO1, made a good point. The HO1 locally at the cellular level has very high levels of carbon monoxide. So if you could simulate that and do it in a very controlled fashion, then you'd have no concern over safety. And that would be, to me, the ideal, particularly if you can get it in the body, as every other medicine that people like to deliver, which is as a pill.
Yes.
For sure.
Yes. Okay, thank you very much, Leo. I think we've learned a lot today about heme oxygenase and CO, and obviously you have this enormous experience that, we are very happy to be able to share with us and to our listeners. We will keep looking around, see your papers coming out, and we will be in touch for perhaps another podcast in the future.
I would welcome that, and I can't thank you enough for the enthusiastic invitation, and I look forward to keeping up with you because your labs and the field has still got a lot of room to grow. So I think your contributions are just as important, and I look forward to reading your papers as much as anything else. So thank you again for having me.
Thank you, Leo.